Hello students, welcome to another lecture on Understanding Biology chapter 23. In this lecture you will learn about viruses. At the end of this lecture you should be able to describe the different structural forms of viruses, distinguish between lytic and lysogenic cycles in bacteriophage, describe how the HIV virus infect human cells and explain why new influenza strains arise periodically. So, when you think about viruses, what do you think? Like, are they alive? Are they not alive? Or are they active or inactive? So, viruses are actually considered active or inactive, both in the same time. Actually, they are also not considered as organisms, because viruses actually lack many of the features considered with life. These include cellular structure and independent metabolism or independent replication. Even though they are not considered as organisms, because of their disease producing potential, viruses are important biological agents. So they are very well adapted pathogens, cellular pathogens. Therefore, they are very important for us to understand and study and use that knowledge to cure some of the diseases caused by these viruses. So, they are actually considered as the most successful pathogens or infectious agents of all the time. So, all viruses have some basic structure. Together, when you look at viruses, they have a core of nucleic acid protected by a protein coat. This structure actually lack a cytoplasm. So individual virus only contain a single type of nucleic acid. It could be either DNA or RNA. This DNA or RNA genome can either be circular or linear, single or double-stranded. Nearly all viruses form a coat protein, but some viruses that do not produce coat protein also have been discovered before. So I told you before that viruses are not organisms, but I also told you that they could always be active, considered to be active or inactive in the same time. So let's look at why. So when you think about a virus, they need a host cell all the time. They are obligate parasites, which means that without a host, they cannot carry out any of the replication and production of new virion particles. So outside the host cell, the inactive form of the virus is called a virion. It is not either alive or dead, just inactive. So we learned that earlier that almost all of the viruses contain a coat protein that protects its nucleic acid core. This protein coat is made out of repeating units of few protein subunits coated by a viral genome itself. Some viruses store specialized enzymes in the nucleic acid core that will aid them to a special function, such as entering the cell or exiting the cell, or also replicate their own genome. One such enzyme is reverse transcriptase. You might have heard about this enzyme. This enzyme allow converting RNA into DNA, which is not found in host cell. Many animal viruses, on the other hand, contain another layer on top of the protein core. This layer is called an envelope. This envelope is derived from host cell membrane and is added when virus is exciting from the host cell by lysing it. So viruses, I told you that they are obligate intracellular parasite. They have been discovered in every kind of organism investigated. Viruses can infect many different types of organisms. Their host range can vary depending on the virus. So type of the organism virus can infect is called its host range. And each type of virus could have limited host range, but there are some other viruses that have very broad host ranges. They are known as generalist virus. One such example of a generalist virus is cucumber mosaic virus. They has very broad host range. That is one of the viruses that is known to have a large host range that it can infect. So virus may also have tissue tropism, which means that they can only infect certain type of tissue, and virus can also enter their host genome and remain dormant or latent for years. So there are more kinds of viruses existing than organisms, so they're actually very problematic cause for humans because of the disease causing viruses. One such pandemic that occurred recently, as you all know, COVID pandemic, which is caused by COVID virus. So when you consider virus shapes, most viruses can be divided into two simple shapes. It can either be helical, which is a rod-shaped structure, or icosahedral, which is a dome shape that maximize the internal capacity. But there are some viruses such as T-even bacteriophage that have complex structure. This bacteriophage contain icosahedral head and a helical axis. Large animal viruses such as flu virus can have regular but polymorphic shapes. So here is a picture actually showing sizes and shapes of viruses. So you actually can get a comparison between two different very well known bacteria such as E. coli and Streptococcus. Comparing these two bacteria to viruses, you can see that virus are very small in size. And here is a eukaryotic yeast cell and largest cell there, and then different kinds of viruses, herpes virus, rabies virus, HIV, influenza. So you can see different helical shape, icosahedral shape, and T2 bacteriophage which has both helical and icosahedral structure. So virus can be reproduced only when they enter their host cell. As you know, they are called obligate internal parasites. So virus is simply a set of instructions encoded in their genome that can trick the host cell, to use the host cell as a machinery for their own replication and metabolic enzymes of the host cell into making copies of the virus. Therefore, outside of the cell, virion is actually called metabolically inert or not functioning. So viruses lack their own ribosomes and enzymes for protein synthesis and nucleic acid synthesis. Therefore, viruses hijack the cell's transcription and translation machinery to produce virus proteins. They actually can be encoded as early, middle, and late genes, which produce different kinds of proteins that is necessary for replicating their genome, making them more protein, and assembling the virus. So when you consider virus genomes, we know the virus can have one different kind of nucleic acid. There are great variation in type of nucleic acid and number of strands they have. Most RNA viruses, for an example, have single strand. Their RNA is replicated and assembled in the cytosol of the host cell. Then RNA genome replication is considered error prone, because we all know now that RNA polymerase doesn't contain proofreading activity like DNA polymerase. This actually leads to higher rate of mutation, which makes it difficult for target by host immune cells and the vaccines that we make and different kinds of drugs. Therefore, we consider that it is really difficult to completely treat a viral infection. When you consider single-stranded RNA virus genome, they can be positive strand or negative strand RNA viruses. Positive strand RNA viruses contain a genome that has the same base sequences as the mRNA. So when this positive strand RNA virus enter a host cell, and release their RNA genome, it can immediately produce proteins because it's similar to mRNA. Therefore, a positive strand RNA virus actually serving as mRNA. On the other hand, we have also negative strand RNA viruses. Their viral RNA genome is complementary to the viral mRNA. And we also have a special class of RNA viruses called retroviruses. They have an RNA genome that is reverse transcribed into DNA by the enzyme reverse transcriptase that we talked about earlier. And DNA viruses, on the other hand, have double-stranded genome. This DNA is replicated in the nucleus of the eukaryotic host cell. Now that we talked about eukaryotic viruses, let's us focus on another category of viruses that infect bacteria. These viruses are actually called bacteriophages. These viruses are very diverse from each other and only united by a bacterial host. They actually only infect bacterial host. They are called phages for short. E. coli infecting viruses are the most studied so far. E. coli infecting viruses are best studies because these viruses are very common, and there are other viruses that are infecting archaea as well. These are different from bacterial viruses. So the E. coli infecting viruses that is best studies are called T series. We have T1, T2, T3 bacteriophages that we studied extensively. So I told you that viruses that infecting archaea is a little bit different compared to viruses that infecting bacteria. So bacteria viruses exhibit two reproductive cycles, lytic and lysogenic cycles. It's important that you understand the difference between lytic and lysogenic cycles. So let's look at them individually. During the lytic cycle, the first virus is attached to the host cell. This step limits the host range of the virus because the attachment is required. So for attachment of the virus, they require a specific protein on the surface of the host cell. After attachment, virus penetrates to the host cell, which results the release of the viral genome to the host. Once inside the host cell, during the synthesis phase, virus take over host cells, replication and protein synthesis machinery, and synthesize viral genome and new viruses. And then these are actually divided into several phases such as attachment phase, penetration or injection, and synthesis phase. After synthesis, they assemble their viral protein and the nucleic acid into the virion particle, and then they get released from the bacterial cells by lysing the cell wall of the bacteria. So mature virus particles will be released through the enzyme that lyses the host or budding through the host cell wall. And this is the life cycle of lytic. A lytic life cycle usually kill the bacterial cell in the release phase because they lyse the bacterial cell wall. So we call eclipse period the time between the absorption and the formation of new viral particles. If the cell is lysed at this point, if any active virions are released, this is the time that the lytic cycle is completed and the bacterial host is killed. On the other hand, the lysogenic life cycle of bacteriophages is a little bit different. They are capable of latent infection. Latent infection means that they do not usually kill the host cell. They do this by integrating their genome into the host genome. This integration allows the virus to replicate along with the host cell. And this type of viruses are called temperate or lysogenic viruses. And this DNA segment that is integrated to the host genome is now called a prophage. So when we look at the lysogenic bacteriophage life cycle, the best studied lysogenic bacteriophage is called phage lambda in E. coli. And one of the interesting facts about phage lambda is that the early events of lambda infecting a host cell constitute a genetic switch that will determine whether the virus will follow the lysogenic or lytic reproductive cycle. When a stress situation occurs, these pro-pages that is inserted into the host genome can actually switch back into the lytic life cycle and cut them off from there and lyse the cell and release new virion particles. So this is actually involving controlling the gene expression pattern of this page lambda, which is very interesting. So I have a picture here, figure 23.17, which showing both the lytic cycle and lysogenic cycle. As you can see, in the lytic cycle, after the assembly, the virus new virion particles are released by lysing the cell. It can also go out by budding. And in lysogenic life cycle, you can see this virus genome is integrated into the host genome, and it reproduces or replicate with the host cell. So now we are actually taking some examples to understand why viruses are very important for humans. So we actually, one of the viral diseases we know very much is HIV. There are a wide variety of animal viruses here in infecting humans. So we are going to look at some of the virus diseases that infecting humans. One of the popular diseases and one of the dangerous diseases caused by virus HIV is called AIDS. And AIDS was first reported in US in 1981 and known to be caused by human immunodeficiency virus or HIV. It is shown that the origin of HIV was in Africa, dating back to 1950s. Infected humans have varying degrees of resistance to HIV, which is very interesting now that we are understanding about this virus. So why HIV is such a dangerous disease for susceptible individuals? This is because HIV actually targets cells that are critical to the human immune response. Immune cells are characterized based on the protein displayed on their surfaces. HIV targets cells that express the antigen CD4. So these cells that actually display the antigen CD4 are called CD4 plus cells. These specific cells infected by HIV are T helper cells, which is very important to regulate human immune responses. Without these CD4 plus cells, the body cannot maintain the defense against the invading pathogens that can usually a healthy individual can fight off. So when a susceptible person has HIV infection, normal common cold or common flu can kill them because their immune system is not functioning very well. So therefore, it is very dangerous disease for susceptible individuals. And also one of the important facts about HIV is that when HIV infection happens, the clinical symptoms do not begin until after a long latency period, usually about 8 to 10 years. So you wouldn't know if you have a HIV infection because clinical symptoms will not appear for such a long latent period, such as 8 to 10 years. So when you look at the HIV infection cycle, it is good to understand how HIV is infecting. So it is also a good model to understand how animal virus replicates inside the cell. When HIV is introduced to the human bloodstream, virus will circulate to the bloodstream and infect only CD4 plus cells. Virus will recognize the CD4 plus cells by binding to the glycoprotein 120 on the virion cell surface, precisely fitting into the cell surface marker of CD4 cell, macrophages, and other white blood cells. There are also several co-receptors such as CCR5 receptor could be another pathway for HIV entry. After docking into the CD4 plus receptor, HIV requires the CCR5 co-receptor to pull itself across the cell membrane to go into the cell, CD4 cell. After inside the cell, HIV will shed their protective coat, and then virus RNA in the host cytoplasm will use reverse transcriptase to synthesize double-stranded DNA copy of its viral RNA, which then enter to the nucleus and incorporate itself into the host genome. After variable period of dormancy, they actually HIV provirus can direct the host machinery to produce more copies of this virus. Then they will this envelope virus will lyse the host cell, and then they will go out to the bloodstream again and start another infection cycle, killing off these important immune cells. So this is why it is challenging to treat the HIV, because they can actually mutate during the latent period, and they can undergo evolution and produce a different strains, and they can actually infect the cell again and again, killing off our important cells like CD4 cells. So here is a picture actually showing the attachment, entry, replication and assembly, and then release of the virus cell. And this is typically how animal viruses are replicating inside host cells. Another important human virus we consider here is influenza virus. It is considered to be the most lethal pathogen in human history. Flu viruses are actually segmented RNA viruses that contain 11 genes in 8 genomic segments. They are enveloped animal viruses that look like a spherical, which studded with the spikes. These spikes actually contain two types of protein, hemagglutinin that allow the virus to enter respiratory cell, and neuraminidase that allow them to escape. Based on the composition of the H and N, flu viruses are called subtypes. So there are subtypes based on the hemagglutinin and neuraminidase. The composition of these two proteins, we can actually categorize these flu viruses into subtypes. So those H and N molecules can accumulate mutations. Therefore, we cannot target same H and N molecules for varying different kinds of viruses. Therefore, we have to have yearly flu vaccine. Yearly flu vaccines, actually what they do is they predict mutations in these H and N molecules and synthesize vaccines based on their prediction. Currently, there are flu viruses are currently classified into 13 H subtypes and 9 H-N subtypes. They are given names such as H1N1. Each subtype is required different vaccine, therefore. Flu viruses can undergo genetic recombination, therefore it is very difficult to control or predict or have the same flu vaccine yearly. They have to come up with the different flu vaccine by predicting these different mutations that happen in their RNA genome. So now that we know that they actually change their genome, each time that they change their genome, we have this thing called antigenic shift. Due to RNA polymerase that lack of the proofreading ability, genome replication lead to high mutation rates. When mutation occur in either H or N portion, we call this antigenic drift. Antigenic drift make vaccine ineffective against new mutations. So there are several antigenic shifts happened previously that caused pandemics, such as Spanish flu in 1918, Asian flu in 1957, Hong Kong flu in 1968, and finally we all know about COVID-19 that has not undergone an antigenic shift as it is not an influenza. So in summary, viruses have very simple structure that include nucleic acid core protected by a protein shell. Viruses are obligate parasites that contain diverse genomes which could either be DNA or RNA. Bacteriophages can have two major types of life cycles, lytic or lysogenic. HIV is a retrovirus that enters the cell via membrane fusion and primary infect immune cell CD4+, and HIV can compromise the host immune system, and influenza, perhaps the deadliest virus to humans, has given rise to pandemics. That is the end of the lecture, and I hope you enjoy this lecture. Until we meet with another lecture, have a good day. Thank you.